Okay, so I'm going to basically just run the lab without comment. I will follow the step-by-step directions given in the data and observations. So as you watch the video, you will need to follow along in your data and observations and record the data so that you can hand the lab in to me. First thing we're going to do is select the wing size. You record the genotypes and the phenotypes. Antenna. Color. And dots. And as the instructions say, we will do wing edge at a later time during the experiment. Answer question 6. And we mate. And the results. Here you can see it is the F1 generation. So be sure that you are reading along on the data and observation sheet as you are watching this, because you should be answering your questions as we go through. So you should be through question number 10 at this point. Now we're going to take two F2 and mate them. You can see this is generation F2. Actually, I misspoke. We took two F1s, mated to get the F2 generation. Okay, so now we're going to run the monohybrid crosses that you see on table 2 of your data sheet. We have already done big B by big B, big B. So now we will do little b, little b, little b, little b. And we'll take our F1s. See what we get with F2. Now we will take big B, little b, mate. And again, take F1 to see what we get with F2. So be sure you are recording your data and observations on your sheets. Now experiment 2. And we do not carry through to the F2 generation. We are only observing the F1 generation in our experiment 2, table 2. Little b, big B, little b. And again, only recording data for F1, not F2. And finally, we're going to look at experiment 3, where we look at dots. F1 generation. Now take 2 of the F1 generation and make them to give us the F2 generation. Okay, now we are starting on determining the genotype from the phenotype. That will be M on your data and observation sheets. We're going to click Hide Genotype. We're going to select wing edge. Okay, so you now would fill in the rest of Table 1, answering the questions 15 through 17 on your data and observation sheets. Our first cross, we will do a round wing, F1 generation, take 1 from each of the F1 generation, and observe your F2 generation. This will be recorded in Table 4, and then we will continue to carry out the matings. Listed in Table 4. So now pointed wing behind pointed wing. Take 2 F1s. Give the F2. Now round wing, pointed ring, two of the F1s, give us F2. So that should enable you to fill out Table 4, this set of experiments. Okay, so for part 3, we're going to discover a genotype. We have unknown number 8. You can determine some of the traits by just observing this one's phenotype. And now we'll run a couple crosses to verify. That's what you're going to get with antenna, color, wing edge, and then dots. So fill in Table 5 with this. This concludes all the data and observation sheet. View the analysis and conclusion sheet to fill out Table 3. This will be a separate video.